Welcome and thank you for joining us today for the webinar, QuantaSoft Software, Learn to Analyze Multiplex Data. Our goal is to provide you with a few simple steps to successfully and accurately analyze duplex ddPCR data by utilizing various thresholding and cluster calling techniques. QuantaSoft makes duplex data analysis easy with the use of 2D views. First, I'd like to introduce myself. My name is Leanne Heisentreit. For the past year, I have been a member of the Bio-Rad technical support team, and I am a specialist for the ddPCR products. I hope these tips and tricks for successful data analysis will be useful for your own research. Before we get started, let's quickly review 3 useful webinar features. If you want to increase the size of your display window, click the full screen icon on the right side of the upper margin next to the word Welcome. You may notice that your microphones are muted. We've done this to ensure webinar clarity for everyone. Lastly, be sure to submit any questions throughout this webinar by using the Enter Questions chat window. At the end, we will address as many as possible. If we do not have time to answer all of them, then we will attempt to contact you with answers in the next few days. Today we will cover how to analyze multiplex data in the QuantaSoft software. First, we will take a look at various types of multiplex assays and show examples of ideal and challenging data sets. Then we'll move into the software to demonstrate the various thresholding options. When using a Bio-Rad ddPCR system, multiplex assays can be divided into 2 types. The first are assays that examine a single target per channel with the use of FAM-hex or FAM-vic fluorophore combinations. When examining a single target per channel, independent assays are one possible design option. Independent assays have 2 primer and probe sets within a single well, where each primer and probe amplifies and identifies a unique gene target. Examples of this design type include copy number variation, rare sequence detection, as well as 2 target gene expression assays. In contrast, dependent assays have a single primer pair with 2 competitive probes. An example of this type of assay would be a rare mutation detection assay, which are also known as SNP assays, or our library quantification kits for next-gen sequencing. The other type of multiplexing that is possible on the Bio-Rad ddPCR system are assays that examine 2 or more targets per channel. For example, using EvaGreen chemistry, which is a double-stranded DNA binding dye, multiplexing in a single well is possible by altering various assay components to achieve cluster separation. This can be done by differing primer concentrations, amplicon size, or the TM of the primers. This example on the right, we have 2 primer sets that were placed into a single well, one that was designed with a TM of 59 degrees, the other with a TM of 63, and the anneal extension step for the PCR reaction was done at 63 degrees. This resulted in a lower overall amplitude for the primer set number 2. Multiplexing can also be done with probe assays by varying primer concentrations. This example on the right is a 3-plex assay where we have a single probe in the FAM channel and 2 in the hex, one of which was added at a lower overall concentration than the other. Because of the complex nature of assay design and data analysis for detecting more than 1 target per channel, these are considered advanced ddPCR applications that will be covered in future webinars. In the QuantaSoft software, you can visualize your data by using either the 1D or the 2D amplitude views when you have 2 targets enabled on the plate setup for a sample well. When selecting a threshold in QuantaSoft with multiplex data, it is helpful to utilize the 2D amplitude view. With this data in the 2D view, it is easy to see the 4 distinct cluster populations. We have our double negatives, our single positives, as well as our double positive droplets. However, in the 1D view, it is nearly impossible to determine the double positive droplet population. In this case, where the droplet clusters are not at perfect right angles to one another, it will appear as though you have double bands in one or more of your channels. When the clusters are at perfect right angles to one another, we refer to that type of data as orthogonal. This double banding pattern is the result of viewing two-dimensional data in one dimension. In other words, it is as if you were looking at a 2D graph from the edge, where one dimension is visible and the other is not. In this case, the orange cluster and the green cluster are slightly offset in the 2D view, whereas the blue and the orange are not. The data on the previous slide had nice distinct clusters, which allowed us to easily determine which droplets belonged in the four populations. However, as you can see here, the data sets are not always well defined. This 2D data is a probe assay that contains rain, which is also otherwise known as droplets that have an amplitude outside of the positive and negative clusters. In this instance, it is what we refer to as middle rain, since the droplet amplitudes fall between the middle of the negative and the positive clusters. Middle rain is typically the result of differences in reaction efficiency, where all droplets do not have the same amount of PCR product at the end of thermal cycling. It is also characteristics of certain types of template, such as DNA extracted from FFP tissues or cDNA. Another common type of rain is what we refer to as negative rain, or droplets that fall below the negative population. This droplet population has reduced fluorescence due to droplet damage. In the case of droplet damage, you will also see droplets that fall within the middle region. Therefore, the main way to distinguish between damage and reaction inefficiency would be to look for droplets that trail off the negatives, such as shown here. Shortly, we'll move into the software to examine how rain can affect thresholding and then possibly concentration calls. Here is an example of typical data from a SNP, or rare mutation detection assay. As you can see, this assay type is lacking orthogonality, since the clusters are not at 90-degree angles to one another. In a SNP assay, clusters are commonly deviated from the corners due to probe cross-reactivity. In this case, the double positives are located at a diagonal between the two single positive populations, rather than in the upper right-hand corner where we would expect them to be if it was orthogonal data. This shift is due to varying amounts of template in the individual droplets, along with probe cross-reactivity, and is normal and expected for SNP assays. In this example here, we can see that our double positive population actually falls out into three distinct clusters. And this is due to having varying amounts of mutant and wild type transcripts within each droplet. This cluster here, we have one wild type and two mutant, one wild type and one mutant, and two wild type and one mutant. Here is a great example of non-orthogonal data that requires unique thresholding. These are typical results from Bio-Rad's ddPCR library quantification kit for Illumina TrueSeq. This kit provides highly precise measurements to determine library concentration and assess library quality prior to sequencing. To do this, the library quantification kit utilizes a single primer pair that flanks a library insert. And it has two unique probes that are specific to the P5 and P7 adapter regions of the TrueSeq Y adapters. This resulting data is unique in that the targets of interest will actually fall along the diagonal, moving away from the double negative population. We can also visualize adapter-adapter ligations, as well as library fragments that contain 3 or more adapter ligations. So let's move into the software so I can demonstrate how you may threshold some of these different types of data sets. So for today's demonstration, I already have some data files open. However, in QuantaSoft, after you load one of your QLP data files from the load option on the plate screen here, you'll be brought to the plate setup. Next, you'll want to click on the Analyze tab. This will bring you to the default concentration graph. From looking at the chart above, we can see that this is a copy number variation experiment. In order to set the thresholding or see the thresholding, we need to click on the 2D amplitude view. So QuantaSoft will automatically threshold data if it can confidently identify 4 distinct clusters in a duplex assay. In this data file, QuantaSoft easily identified the 4 distinct populations. So we have our double negatives, our single positives, and our double positive droplet population. The software will also display an OK status in the chart above if it's able to perform the auto-analysis. It'll also give you a concentration call. Since this is clean orthogonal data with minimal rain, the auto-analysis was easily performed by the software. However, you are still able to change the cluster calls if you want to see the difference or if you disagree with the software. To do this, you have a couple of options. The first of which is the crosshair. As you can see here, when we move over the graph, we are able to click anywhere within that graph and set a new threshold. We can also move it around by simply dragging and dropping to wherever we may want to set the threshold. There's also another very helpful tool for thresholding, and that is the lasso tool shown here. Once you click on the lasso, it'll activate the 4 buttons below. This will allow you to circle or highlight individual droplets you want to include into different clusters. So for example, if we wanted to include these 2 blue droplets as a double positive, we would simply make sure our lasso tool is highlighted, click on the appropriate button below for the double positives, and then circle those droplets. And you can see they have then moved from blue to orange. So now let's take a look at some data that has rain to see how we can use these tools to manually define cluster groups for that type of data. So here is an example of an absolute quantification experiment. This assay has droplets of intermediate fluorescence that fall between the negative and positive population. Since we do not see any trailing off the double negatives, this rain in this example is likely due to varying amounts of endpoint PCR product and not to droplet damage. Even though this assay does contain rain, the software was still able to automatically make the threshold calls, as shown by the OK status and the concentration listed above. However, you may be wondering if the software was correct, so you can move the threshold to see how it affects the concentration calls. So for this one, I'm going to click the crosshair tool, and let's note the concentration above to see how that may change by moving the threshold. So I'm going to set it approximately in the middle. And then as I move the threshold around, the overall concentration does not significantly change. This is because there are only a handful of droplets that are affected out of thousands. Therefore, you don't have to worry about every single droplet being in the correct cluster for this type of data. Next, I want to show you what happens when the software cannot definitively define a threshold for you. In this SNP assay, everything is labeled as a double positive once it's opened. There is also a check status listed above and no call, which notifies us that the software was not able to perform the auto-analysis. However, we can hover over the check for more information. Here it tells us that the automatic analysis was unsuccessful, and that if we'd like to view our concentration, we need to review our data and then use the manual analysis tools. So let's do just that. For this data set, you could use either the crosshair or the lasso tool. So starting with the crosshair, we can simply set the threshold. And now we have our 4 populations highlighted. Alternatively, we could have used the lasso tool to circle those droplets of interest, or we can now include others within a certain population if we like. The last example I'd like to cover today is data from our Illumina TruSeq library quantification kit. Here, our droplets of interest are moving along the diagonal and contain inserts of varying sizes within the library. Fragments will go from larger to smaller as we move away from the negative population. The droplets at the highest point are the adapter-adapter ligations and do not contain an insert. Those that appear above and below are the rare species that have 3 or more adapters ligated into an insert. Here for this data set, we will utilize the lasso function to determine the overall concentration for this library. For this type of data, I find it most useful to call our droplets of interest channel 1 positives, or we'll highlight them with the blue. So those I'm going to circle first. I also then want to call my adapter-adapter ligations in the channel 2 positives. So I'm going to highlight those as well. And I don't see any of the droplets below this diagonal here. And what this allowed us to do was then determine our library concentration. By looking in the graph above, we'll see for channel 1 our library concentration. Another very useful tool for this type of data in the library quantitation is then moving on to the ratio tab. If we click on fractional abundance, it's going to tell us the overall percentage of our library that contains inserts. So for this library sample prep, 98.63% of it contained our droplets of interest. This is a useful tool for not only determining your library concentration, but also the quality, and will therefore allow you to appropriately load and balance your downstream sequencing runs. So just to briefly review what we covered with the software, in order to manually set the threshold for your multiplex data, you first want to click on the Analyze tab, select the 2D amplitude view, and then set the threshold using either the crosshair or the lasso tools, whichever ones make the most sense for your data. I'd like to leave you with a few take-home messages. First, use the 2D plots for analyzing multiplex data. These are going to give you the most information about your data set. QuantaSoft can also automatically call clusters for clean orthogonal data. However, you're also able to manually define the threshold for complex data sets. And lastly, QuantaSoft can calculate the concentration for your next-gen sequencing libraries. All right, I'd like to thank everyone for listening, and now we'll move on to the questions that were submitted throughout the webinar. All right. The first question was on the slide where you indicated that there were 3 different concentrations for wild type and mutant. You indicated that there were 1 mutant and then 2 mutant. Could you clarify this just a bit more? So let's move back to that slide. I believe this was the slide here. All right. So for this one, there are we have our mutant population as well as our double positives as well. These ones fell out into 3 distinct clusters, and based upon the fluorescence of the mutant droplets being higher, you're able to determine that there was 2 mutants in this population here that had overall higher fluorescence, whereas 2 wild types would pull the fluorescence more towards the wild type population. The second question we got was, is there any way to exclude data points in the 2D plot? From what I can tell, data points always have to be called double negative, single positive, or a double positive. And the answer to that one is no. You have to have them to be within one of the 4 groups. Typically, if you want to exclude a droplet, it's best to call it a double negative if you're uncertain or you want to exclude it. The next question we have was, is there any way to filter the data in the event view? For example, rarely something blocks the flow. It would be great to omit those data until the clock clears. That's a very good question. The software automatically will filter some of the data, so there's no way to actually perform that manual filtering. All right. Next question. Can QuantaSoft be downloaded and run on another computer? Absolutely. In order to get a new version of QuantaSoft or another download, simply give a call to us here in tech support and we'd be happy to send you that download link. Next question that came in. Would it be possible to get some information about what the algorithm is for determining the clusters? For this one, the algorithms are actually proprietary, but if you give us a call, we'd be able to give you some general details and go over that a bit further. All right, well, I think that about wraps it up for today. I would like to thank everyone for listening and joining us. And then here is the best way to contact Bio-Rad tech support. So we have our phone number as well as email, and we have a pretty good turnaround time. And for all of those questions that we were unable to get to, we will reach out to you as soon as we can with answers. Thank you for everyone for joining.